Great, well, welcome everybody to the workshop. Really excited to talk about our 3D genomics approach today and some of the exciting results we have in the collaboration with Dr. Snuderl. So in the next 20 minutes or so, I wanted to really cover just two things. The first is I typically assume we're a pretty small company, young company, that you haven't heard of the term 3D genomics. So I'd like to spend first a little bit of time talking about what is 3D genomics, what do we mean by that? And conceptually, how can we use this 3D genomics approach to detect structural variants? On the topic of structural variants, I'll just say that a lot of people use different terms, rearrangements, fusions, translocations, structural variants, etc. I'm just going to stick on the term fusions to sort of mean all of those things. And then probably, what's most exciting and kind of as a primer for Dr. Snuderl's presentation is just a few slides of a summary of how we've applied 3D genomics to identify druggable, diagnostic, and prognostic biomarkers in previously characterized yet driver-negative tumors. A bit of background about Arima. In case you're new to us, you can find more information on our website. But we were spun out of the lab of Dr. Bing Ren back in 2015 at UC San Diego. Launched our first product in 2018, which is a reagent kit pictured there to do this NGS-based 3D genomic profiling. We've raised a couple of financing rounds. We crossed 100 publications from customers last year, and we have a little over 300 customer publications today. We've also launched other products along the way to help you sort of zoom into individual genes, sort of targeted approaches, and expand the sample type compatibility to those that are relevant for clinical use like FFPE samples. Everything that you see here today throughout the workshop you can do yourself with kits, reagents, and companion informatics tools, or send to us for a service. All right, so what is 3D genomics? I was introduced this juxtaposed to linear genomics, which I think most people are more familiar with, is thinking of the genome as a linear string of 3 billion plus bases, A's, T's, C's, and G's, all linearized. Whereas 3D genomics is kind of like using sequencing to sequence the structure of a chromosome. Right? And so this is an NGS-based approach. And in the technique, you preserve the three-dimensional structure of chromosomes, and you read that out using NGS. So it's like sequencing the structure of chromosomes. So you can imagine for a structural variant that changes the structure of a chromosome. If you have a technology that inherently measures the structure using NGS, you can detect changes to that structure. And I have one kind of like workflow slide here. The Arima technology is called Arima IC+, which is really a glorified sort of DNA extraction technique, where a variety of different sample inputs come in: blood, cell lines, frozen tissues, FFPE tissues. And the key thing is that cells in all of these different sample types are intact, and within a cell, the three-dimensional genome is fixed into place, right? So it's not a bunch of linear, chromosomes floating around. It has that three-dimensional structure. And the way that you capture that during sample prep is you fragment the pieces of DNA, and then you ligate them to their spatial neighbors. So this light blue piece here and the dark blue piece here are spatially proximal, but they could originate from very far apart along the genome or the chromosome. And you cut them, and then you ligate them back together. And ultimately, what you want to do is then these molecules that get ligated together, that are part light blue, part dark blue, you want to sequence them, right? Because that'll be essentially one data point from one cell that the light blue piece and the dark blue piece were spatially proximal to each other, and it gives you information about the structure of a chromosome. So once you extract that DNA, it goes through a conventional NGS library prep for Illumina sequencing, and then on the back end, we provide informatics tools to identify fusions from this 3D genomics data. And what our customers and collaborators really sort of like about this approach is that it plugs into the existing NGS ecosystem that are sort of already established in their labs. The informatics is all containerized, so it's portable and scalable, and it provides robust and reliable results. All right, so how do we sort of, I've talked about sort of on the surface what is 3D genomics, well then how do you leverage that data type to detect fusions in cancer specimens? And again, I like to sort of juxtapose the linear genomics approach to the 3D genomics approach. Let's start with a normal cell. You have two different chromosomes here, chromosome A and B. We're thinking in a linear context here, so I have them linearized. And there's no fusion. And so if you were to sequence this normal cell, you would see reads sort of scattered around chromosome A and chromosome B like you see here. In the case of a gene fusion, depicted here, where chromosome A has fused to chromosome B, the key takeaway here is that if you sequence this cell, the only reads that are informative for there being a fusion are the ones that pile up at the breakpoint, the ones that are chimeric or split reads, right? Because you have a little bit of mapping to gene A and a little bit of mapping to gene B at that breakpoint. So those are the informative ones with a linear genomics approach. Now for 3D genomics, in reality, these chromosomes are organized into some three-dimensional structure within the cell. An important detail here is that these two chromosomes are not intermingling with each other, right, which is what happens in healthy cells. And then if I were to project this sort of as a linear representation, these blue dots over here on the left are those spatially proximal points that get ligated together during the sample prep. So here on chromosome A, these blue dots are sort of like these 3D linkages, right, within chromosome A, and over here on chromosome B, these 3D linkages within chromosome B, right? So this is in a healthy state. In the case of a gene fusion, you have a fused chromosome A and B. Now what's different is you have these 3D genome linkages between chromosome A and B. So it's crosstalk between two different chromosomes when there typically is not crosstalk between two different chromosomes in a karyotypically normal cell. And if I project this again into sort of a linear space, here's that fusion, and here are all those 3D linked reads that hop over the breakpoint. All of these are not supposed to be there, and are kind of like the signal that the 3D genome type of approach harvests to help shine a spotlight on these fusions. Now this is just from a single cell, right? So move that image up there. Let's take a second cell. There's going to be another set of linkages between chromosome A and B, cell 3, and so forth. Hundreds of cells, thousands of cells, you get this huge amplification sort of this fusion signal. And the two things I would want to draw your eyes to is if you kind of scan down right here at the breakpoint, there are still reads that are truly at the breakpoint that provide that sort of split read or that typical evidence of a fusion, which is just like sort of the linear genomics approach. And then you have this massive amplification, is what we call it, of signal from all the 3D genome linked reads that hop over the breakpoint that are not there in a linear genomics approach. So here's a schematic paired with some actual data. We did a enrichment of our 3D genomics library where we had probes that pulled down the BCR gene in a sample that we knew had a BCR-enabled fusion. And so what we'd expect to see is that primary evidence at the breakpoint of the reads that have the breakpoint actually in them, right? That's sort of evidence number one. And if you show this sort of in an IGV view, and you look right at the breakpoints, and these are real reads from our experiment, we see 78 reads that are right at the known breakpoint that are split between BCR and enable. But the real sort of advantage here is that we'll have all these other reads where BCR is interacting in that three-dimensional space with the fusion partner and the locus beyond the fusion partner because of the structure of that fused chromosome. So that's what these arcs depict here. And so all of these reads that come from BCR and are 3D linked to something else on this locus are informative. And what that actually looks like in an IGV type of representation is over here on the right, all these pile up of reads from BCR. Each read is linked to one other read on ABLE over here. Right, so we have these 78 reads at the breakpoint, but we also have 7,272 reads that connect BCR to ABLE in 3D space via the structure of the chromosome. Right? That's a 100-fold signal amplification to help you find that fusion. And of course, if we do this in a healthy individual, there's no chimeric reads at the breakpoint, and there's also zero, in this case, zero reads that link BCR to ABLE, showing the specificity of the technique and kind of a feel for the background level. Now, I just have one slide here because I think this is going to be helpful for Dr. Snuderl's presentation, as I've shown you kind of the IGV type of visualization for these data. The data is also often depicted as what's called a heat map. And I just have one slide because I think that'll be helpful to interpret some of the figures in his presentation. So what you're looking at on the left, I'll go through this pretty quickly, is a karyotypically normal sample. And all these red dots represent the number of three-dimensional linkages between any 2 points along the genome. And so if you can see my cursor over here, this is chromosome 1, 2, 3, 4, all the different columns, and chromosome 1, 2, 3, 4 are all the different rows. And so there's lots of that 3D linkage signal within a chromosome. Like I showed you in the schematic, chromosomes tend to form a structure and fold on themselves. So you'd expect lots of those 3D linkages within a chromosome. And this white space over here just basically represents very few linkages between two different chromosomes in a healthy cell. In a karyotypically abnormal sample, this happens to be a colorectal FFPE tumor with a ROS1 fusion. You see these really strong intrachromosomal-like linkages, but they're occurring between two different chromosomes, which really stands out like a sore thumb when you look at these these heat maps, and is the basis of what the algorithms use to try to find a particular fusion. It looks for changes essentially in this pattern. Right, so all of these would be structural variants or fusions. And you can sort of zoom in on these to kind of get gene level resolution, which I think he'll show in his presentation. So a couple, I would say key takeaways so far is using this 3D genomics approach can provide a few key insights. I think the first one, which I'll talk about briefly, is just the ability to detect known fusions. It's not just for finding novel fusions, but it can also find known fusions that are found by other methods like FISH or RNA or DNA sequencing. I'll show you that we can go back in time and profile old archival FFPE specimens from 10 plus years old. And third, we can find fusions that are not detected or missed by other technologies. And so we're just sort of at a high level summary here. So far, the time I made this slide, it's a little outdated now, we have profiled 217 patient samples across 20 different tumor types. The different tumor types are shown here across the x-axis, and then the number of samples is in the y-axis. Most of these are coming from sarcomas or CNS tumors, just sort of due to the nature of the collaboration that we have with Dr. Snuderl and a few other collaborators, but we also have lung, some of the solid hemes like plasmacytomas and lymphomas, GI, breast, and others. In terms of concordance, every sample that we've profiled so far that has been previously characterized by one of these technologies over here on the left, so whether it's an Archer panel, FISH, a comprehensive genomic profiling DNA panel, whole transcriptome, in all 81 of these cases, we can find the fusion that was known to be there. There was actually a couple cases where we were discordant, and then we went back and tested the sample. There was 2 of these, and it turned out to be a false positive on the orthogonal method, actually, to sort of speak to the specificity of the technique. And we presented this at AACR this past year, a collaboration with Children's Mercy Hospital on 5 of these alveolar rhabdomyosarcoma tumors that are archived between 8 and 12 years. Also wanted to point out the amount of DNA that we were able to extract from them and put into our prep was very low. In all cases, we found fusion and the partner, which is either PAX3 or PAX7, whereas the orthogonal method, whether it be break apart fish or fusion fish or karyotyping, could usually only find the first partner here, which is FOXO1. And I think once we started working with Dr. Snuderl, we I think became acutely aware of the problem of patients being tested with really state-of-the-art molecular profiling techniques and still being driver negative, meaning no clinically actionable driver could be detected from these samples. And the exact percentage of these probably depends on who you talk to or what assays are being run. But in this study out of MSK from last year, approximately 54% of patients with advanced cancers were driver negative, and here's the spread across different tumor types, which is approximately, as we estimate, about 400 to 500,000 patients per year with advanced cancers that are driver negative. And part of the reason for this sort of high driver negative rate is technological. I think we're all probably aware of the sort of limitations of FISH in terms of the single gene testing and throughput, and often not revealing the partner. RNA sequencing often does not excel on FFPE blocks. And something I want to spend a little more time talking about because it's relevant to Dr. Snuderl's presentation here too, is that clinical NGS panels look, they're targeting the gene bodies of, clinically actionable genes. And so if there is a breakpoint that is beyond the gene body, something that I will refer to as a proximal fusion, they will be missed because they're not being targeted. Right? But these are still mechanistically and clinically significant. I think the poster child for the significance of these is in lymphoma, right, where MYC, BCL2, and others translocate to the IGH locus. They don't produce a typical gene-to-gene fusion, and instead these oncogenes are activated because they become under the control of IGH locus regulatory elements, such as enhancers. And from this review almost 15 years ago, it was known at that time that here is gene A, right, on one chromosome. Let's call this MYC. Here is gene B. Let's call this the IGH locus, and some of those enhancers here. When these break at the arrows and they fuse together, this MYC gene is juxtaposed to the IGH enhancer here, and that was thought to activate these oncogenes. Okay. And they have clinical utility. If you look at the NCCN guidelines, FISH testing for MYC and BCL2 and other rearrangements is either considered essential or useful in certain circumstances, and has been known to retrospectively stratify overall survival, here being MYC positive rearrangements versus MYC negative rearrangements in lymphomas that have been treated with R-CHOP therapy. This was 14 years ago, I think. And a lot of recent studies have focused on the mechanisms of sort of how these oncogenes are activated from proximal fusion events, some of which fueled by Arima technology, shown here. And while I think it's well accepted and recognized in heme tumors like lymphomas, there's also been reports really focusing, I think, on the observation of proximal fusions in solid tumors and then trying to tease apart those mechanisms. And it kind of makes sense where if you have a proximal fusion with a breakpoint outside of a cancer-driving gene in a solid tumor, and it happens to juxtapose that to a regulatory element that's active on the partner locus, that it could activate that oncogene with a similar mechanism. Right? And that's what's been found. I think over the past 8 years, there's been several papers, some of which also have used Arima technology. And this one here, that's a preprint, was just covered in Drug Discovery News and came out this week. And I think they have emerging clinical utility. They're not written into any NCCN guidelines that I'm aware of, but they do retrospectively stratify patient survival in neuroblastoma, and there's ongoing clinical trials to try to disrupt the regulatory elements in patients that have these proximal fusions around MYC in pediatric brain tumors. So with that being said, it's incredibly important to not only be able to detect the typical gene-to-gene fusion, but also the proximal fusions. We kind of think of our technology as a floodlight, shown here, where all these reads that I showed you that link between gene B and gene A, we are able to see all of them, right, to sort of illuminate a gene-to-gene fusion. Whereas the typical linear genomics, you have the reads at the breakpoint. But in the case of a proximal fusion, which is shown here, where you have the breakpoint over here, so it's not a gene-to-gene fusion, you'll still capture those 3D linkages between the target gene of interest and the partner locus, even if it's not a chimeric gene-to-gene fusion. Right. Whereas the typical approaches, of course, will not be able to see that. So that kind of brings me to my last couple of slides here. We've analyzed in total 140 driver-negative patient tumors. 122 of those were with Dr. Snuderl. And of those 140, we're able to identify a fusion that implicates a biomarker that is the target of an already approved, FDA-approved on-market therapy. So that's about 33% of the time that's what we find. And about 6% of the time we find a fusion biomarker for a drug that's ongoing in trial, and 14% of the time fusion biomarker that has diagnostic or prognostic significance. So that's about 53% at the time that we made this slide. And most of these data comes from 4 different sample types: gynecological sarcomas, solid hemes like plasmacytomas and lymphomas, and brain tumors. And we have this emerging cohort here that are colorectal tumors, and these are driver negative after pretty expansive profiling. These were all previously profiled by Caris and came back driver negative, and we're working our way through these. But so far, 4 out of those 5 we can find a clinically actionable biomarker. Here's just a smattering of the biomarkers themselves, the size of which correlates to the incidence at which we find them. So a lot of these homologous recombination repair targets that predict response to PARP inhibitors, the NTRK genes, FGFR genes, all that you can see here. So in conclusion, shown you that the 3D genomics approach is concordant with existing methods. It has some of the benefits of improved sensitivity, which we think increases the percent targetable by detecting either gene-to-gene fusions that were missed or these proximal fusions that were never going to be detected by a typical DNA or RNA panel. And we think it has greater accessibility because this is all a DNA-based workflow as opposed to RNA-based, and can go back in time on old archival and low-input FFPE. I think Matija will talk a little bit more about some of the prospective cases and the therapeutic value. Just want to acknowledge all of our collaborators. We're very grateful. Shown here, I won't read off all the names, and thank you so much for your attention.